Hello, my name is Duncan Mullins. I am a second-year PhD student in the School of Chemical Engineering at Oklahoma State University, working in Dr. Ashley Ford Versip's lab. This is a poster over my project, a mathematical model of the glomerular filtration barrier damage in diabetic kidney disease. So DKD is a severe medical condition that results in catastrophic damage to the kidneys and is the leading cause of kidney failure, especially in the US. Within the kidney, the glomerulus is the main area of damage within DKD. The glomerulus is a small bundle of capillaries contained within the nephrons, which are the individual functional units of the kidney. So because of this small scale, mathematical modeling is a very powerful tool for doing research in this area. The glomerular filtration barrier is an essential component of the glomerulus, composed here of three layers: the GBM in pink, the endothelial cells in green, specifically the gaps between them, called fenestrations, and the endothelial glycocalyx here in yellow on the underside of the endothelial cells. So damage to the GFB is one of the main sources of albuminuria in DKD, which is the loss of correct function of kidneys when they are allowing protein to escape as well as waste. So this work aims to create a mathematical model to track and estimate damage to the GFB in DKD. Here's kind of a cartoon illustrating a healthy versus damaged GFB. And this is my simulation considerations. I track the levels of three chemicals that are known to be chronically elevated in DKD: TGF-beta, VEGF, which are growth cytokines, and glucose, which is sugar, as well as the effects they have on the GFB. Here you will notice that VEGF also creates a feedback where it continually creates additional TGF-beta production. So a main focus of this work has been combing the literature for in vivo or in vitro data sets, most of them in mice, which presented an additional complication. However, I was able to find a good number and have developed mathematical correlations for those data sets and then combined them. Each J here is a separate estimated correlation parameter to sort of tabulate and combine the damage. And so here you can see the graphs of this parameter estimation versus the data sets from which these correlations were estimated, and they are pretty decent fits, albeit not perfect. So the simulation is also able to track the concentration of those three aforementioned chemicals within the GFB over the course of DKD. Here you will notice the effects of that feedback loop I mentioned, where TGF-beta is significantly elevated more than the other two. This work has been able to tabulate those damages to each layer, and they have a relatively correct scope and shape, however not a correct timescale, which I'll mention again in a moment. So future work is to validate the model with data sets. You would notice on the last one I have located a data set and begun comparing it. However, the timescales are vastly different. The data set takes place over years or decades, which is physiologically as expected, whereas the current version of the model only takes place over 24 hours. That is a current weakness that I am working to correct. I also want to perform a sensitivity analysis determining the impacts of each estimated parameter and seeing if one of them is significantly larger than the others. Thank you for your time.